You take a standard antihistamine for seasonal allergies and wash it down with a glass of orange juice. Instead of relieving your symptoms, the juice chemically intercepts the medication in your gut, preventing it from ever reaching your bloodstream. The sweet orange contains a specific biological chemistry capable of actively manipulating the borders of human cells. This fruit, Citrus sinensis, is not a naturally occurring species found in the wild. It is a genetically complex hybrid cultivated through ancient agricultural selection. Genetic sequencing traces its maternal DNA back to the Pummelo, a massive citrus variety defined by its notably thick, heavy rind. Its paternal DNA comes from the Mandarin, which contributed the smaller size, sweeter profile, and thin skin we recognize today. This genetic fusion produced a specialized phytochemical profile that targets specific human cellular pathways. Today, sweet oranges dominate 48% of the global citrus market, with millions of tons harvested and processed annually. Our consumption of this crop follows a strict anatomical divide. We extract and drink the inner juice, but we typically discard the outer colored flavido and the white inner albedo, the layers that form the peel. The inner juice and pulp deliver a standard nutritional profile of water, vitamin C, folic acid, and pectin fiber. But the fruit also contains a powerful class of plant compounds called flavonoids hidden within its different structures. Exactly where a flavonoid is located within the orange determines its role, whether it serves as everyday nutrition or a potent pharmacological disruptor. Inside the consumed juice, the most highly concentrated flavonoid is a compound called hesperidin. To understand hesperidin, you have to look at the lining of the human intestine. These cell walls contain organic anion-transporting polypeptides, or OATPs. Specifically, OATP1A2 acts as a cellular door that pulls pharmaceutical drugs out of the gut and into the bloodstream. Hesperidin acts as a competitive inhibitor. It physically binds to these structures, jamming the entrance. When you take an antihistamine like fexofenadine, the drug molecules bounce off the blocked transporters and fail to enter your system. This chart shows how juice volume directly impacts drug absorption. Taking fexofenadine with 300 milliliters of orange juice causes the drug's absorption to drop to just 58%. At 1,200 milliliters of juice, drug absorption plummets to 36%, rendering the medication effectively useless. While orange juice temporarily blocks absorption, it works differently than grapefruit juice. Grapefruit contains compounds that destroy metabolic enzymes, which can lead to dangerous toxic drug accumulations rather than a simple lack of absorption. A single glass of juice creates a targeted temporary chemical blockade of the body's primary pharmaceutical transport system. The juice is only half the story. Industrial processing separates the liquid from the peel, pulp, and seeds, generating orange peel waste, about 50% of the fruit's total weight. This discarded peel layer contains almost all of the fruit's polymethoxylated flavons, or PMFs, specifically Nobiletin and Tangeretin. PMFs are highly lipophilic, or fat-soluble. This allows them to slip through biological borders that block other compounds, moving directly through the tight cellular junctions of the blood-brain barrier into the brain tissue. In laboratory models, these PMFs reduce the abnormal accumulation of neurotoxic amyloid beta peptides, a key factor in neurodegenerative decline. They also inhibit the hyperphosphorylation of tau proteins and have been shown to reverse specific receptor hypofunction. These results indicate that the compounds currently treated as industrial waste are some of the most promising candidates for preventing Alzheimer's and Parkinson's diseases. There is a clinical reality to consider. The drug-blocking interactions in the juice are proven in human trials, but the neuroprotective effects of the peel are currently observed only in cell and animal models. You cannot achieve a therapeutic dose of Nobiletin by simply eating whole oranges. Reaching neuroprotective levels requires the single concentrated dose found in pharmaceutical extracts. This is why juice factories are transitioning into biorefineries. They are engineering ways to salvage PMFs, D-limonene, and pectin from the waste peel to create targeted therapeutic supplements. The sweet orange operates as a dual-action biological system. While its juice temporarily disables pharmaceutical transport, the discarded peel provides the lipophilic molecules currently being studied to defend the aging human brain.